So I'm going to talk a little bit about the embryonic development of HSC and potential therapeutic applications of that. So hematopoietic stem cell transplantation has been and remains an important treatment modality for aggressive forms of leukemias, for marrow failure syndromes that can predispose to leukemia, and to other inherited and acquired blood and immune disorders. Despite decades of significant advancements in hematopoietic stem cell transplantation, including the use of cord blood stem cells, there are still many barriers to optimizing this form of therapy, including the lack of suitable donors for all patients who might benefit from a transplant, and side effects from allogeneic stem cell transplants, such as graft versus host disease. So one of the objectives of research in our lab and the focus of the Hyundai Hope Grant is to use cues from the embryology of HSC to devise new ways to generate HSC that can potentially be used for therapeutics and other applications, including from novel sources, which include pluripotent stem cells. And so these are cells that have been reprogrammed from adult somatic cells to resemble an embryonic stem cell-like state, because they can be essentially expanded in culture indefinitely and can give rise to all types of adult blood cells, all types of adult cells, including blood cells. They represent a potentially limitless source of HSC to increase availability for therapeutic transplantation, but also for disease modeling and for drug discovery. Their properties also make them potentially amenable to homologous gene editing for safer forms of gene therapy. So applications would include use of patient-specific autologous sources for genetic diseases and the addition of allogeneic sources in the cases of leukemias and malignancies. Pluripotent stem cells could be gene-edited to correct genetic mutations, to alter immune properties such as MHC, to limit GVH or enhance GVL effects, or to introduce targeted therapies such as CARs. Ultimately, then, re-derivation of HSC from pluripotent stem cells would allow the transplantation of edited HSC back to the patient. And while there are a number of safety and technical barriers to bringing these types of cells to the clinic, one of the most significant biological barriers has been that current methods, although they're able to generate a variety of blood cells from this type of stem cell, currently lack the ability to generate efficiently engrafting HSC that we see in the bone marrow or in cord blood. And so one approach our lab has taken to overcome this obstacle is to go back to the embryo and ask about the vascular niche requirements for HSC generation. So it turns out that the first hematopoietic stem cells develop in the context of endothelial cells in the dorsal aorta, in a region known as the AGM for aorta gonad mesonephros, and subsequently home to the fetal liver, where they interact with endothelial cells of the liver and undergo further maturation and expansion, suggesting that critical niche signals in these microenvironments are important for the generation and expansion of HSC from embryonic precursors. And to study this, we've used methods from our collaborators in the lab of Shaheen Rafi, which enable the study of organ-specific endothelial cells ex vivo to test the capacity of in vitro vascular niche models from the AGM and fetal liver in mice to determine whether these sources can enhance the generation and expansion of HSC. And we've determined in mouse models that both AGM and fetal liver endothelial cells in co-culture can significantly enhance the ability to generate long-term engrafting hematopoietic stem cells from precursors isolated from embryonic stage mouse embryos. So these studies suggest that the endothelial cells from these sources provide the critical niche signals for both the generation and expansion of HSC. To build upon these studies in the Hope Scholar Grant, we propose to identify additional sources of endothelial cells that support the specific stage, generation and expansion of hematopoietic stem cells in vitro, to evaluate the capacity for these endothelial cells to support generation of HSC from mouse and human pluripotent stem cell models, and to use transcriptional profiling to identify signal pathways that may be critical within these endothelial cells that are necessary and sufficient for HSC generation and expansion. One such pathway that our lab has studied extensively is that of the Notch pathway, where Notch ligands on the surface of endothelial cells activate Notch receptors on hematopoietic stem and progenitor cells, and is involved in both the initial induction or generation of HSC, but also in maintaining HSC and progenitors by limiting their differentiation. And my mentoring lab has previously used Notch ligands to activate Notch signaling in hematopoietic stem and progenitor cells. And this has been used clinically in the context of cord blood to expand progenitors for transplantation therapies. So one of the ultimate goals of this research is to bring some of the factors that we identify from the vascular niche into this system to eventually generate a xeno-free culture system for the generation and expansion of HSC from pluripotent stem cells. And ultimately bring this back to engineer a system in which clinical grade HSC can be generated from pluripotent stem cells for a variety of therapeutic purposes and other research purposes. So I'd like to thank the mentoring lab, my mentor of Bernstein and members of his lab, our collaborators, in particular Shaheen Rafi's lab, who've worked with us with the vascular niche models and funding sources, and of course in particular the Hyundai Hope on Wheels Foundation, whose support will help keep this project going. Thank you.